To perform electrophysiological recordings in Drosophila embryos, they must first be harvested, dechorionated, and dissected. Controlled dissection is made possible with water polymerizing surgical glue, which stabilizes the embryos to cover slips. The internal organs are then removed from stabilized embryos to expose the ventral CNS, the peripheral nerves, and the somatic musculature.
Hi, I'm Dave Featherstone from the Department of Biological Sciences here at the University of Illinois at Chicago.
Hi, I'm Kaiyun Chen from the lab of Dave Featherstone in the Department of Biological Sciences at the University of Illinois at Chicago.
Today we're going to show you how to collect and manually dissect late-stage embryos from Drosophila.
We use this procedure in our lab to prepare for electrophysiological testing of Drosophila neuromuscular system development and function, which I will show you in another upcoming JoVE presentation.
So let's get started.
This protocol begins with embryo collection, a common procedure used in a variety of Drosophila experiments, which we will now review. Establish a mix of 20 to 40 young male and female flies in a laying pot. House them in a humidified chamber at 25 degrees Celsius. To accurately estimate the developmental stage of the embryo, the temperature and humidity should be consistent. Although commercial laying pots are available, they can be constructed from perforated 1